Hello everyone. You are watching Gray's Gyan, where Gray's anatomy is explained in a simple and interesting way. Today's topic is cell surface, apical differentiations. Let's start with a basic question. What exactly are these apical differentiations? Our cells are not just specialized on the inside, their outer surface is also specialized. Cells form special projections on their surface, structures that stick out, and each type serves a different function. There are three major types. First, flagella. The job of flagella is to move the cell itself. The best example is spermatozoa. They have a single flagellum that is 70 micrometers long, which makes them swim. Second, cilia. The job of cilia is to move fluid across the apical surface of the cell, for example, clearing mucus from the respiratory tract. There are two types, motile and primary. We will cover both in detail. Third, microvilli. These increase the surface area so that more absorption can happen, like the brush border of the intestine. One important extra point, in non-dividing epithelial cells, a primary cilium is formed from a centriole-derived basal body, and this has a mechanosensory role. Remember this. Now let us understand the structure, and this part is very important because it is directly asked in exams. A cilium or flagellum has essentially three parts. 1. The shaft, which is 0.25 micrometers in diameter. 2. The tapering tip at the top. And 3. The basal body, at the bottom within the cell's cytoplasm. One important point, the entire structure of a cilium is covered by the plasma membrane, except at the junction at the base. Now the most important part, the axoneme. This is the core of the cilium. It consists of 9 outer microtubule doublets arranged in a circle, with 2 central microtubules in the middle. This is called the 9+2 arrangement. You must remember this answer. Now the components of the axoneme. Radial spokes extend from the outer doublets toward the central pair. Dynean arms are the most important. There are 2 rows of arms on the A subfiber. The A subfiber has 13 protofilaments, and the B subfiber has 10 to 11. The ADPase activity of dynein produces the bending motion. Then there are nexin filaments, which link adjacent doublets together. And tectins, these are scaffolding proteins that extend along the axonal microtubules. Now we are looking at the actual figure from Gray's Anatomy, figure 1.26a. On the left side is the transverse section. This cross-section clearly shows the 9+2 arrangement. In the middle are the 2 circles representing the central microtubules, and surrounding them are the 9 doublets. This is the classic structure. In the longitudinal section, that is, the cilium cut along its length, you can clearly see transport happening from bottom to top. Now pay attention to the callouts on the right side. IFT proteins, these carry tubulin to the ciliary tip. Kinesin 2 motor, this drives anterograde transport, meaning from base to tip. Cytoplasmic dynein 2 motor. This drives retrograde transport, meaning from tip to base. The basal body is visible at the bottom with the BB label. It is centriole-derived and anchors the cilium. And one very important structure, the BB sum. The full form of BB sum is Bardet-Biedl syndrome proteins. These bind to retrograde IFT and facilitate lateral transport of cargoes through the porous barrier of the distal appendage. If Bardet-Biedl syndrome comes up in your exam, remember BB sum. Let us broadly divide cilia into 2 types, primary cilia and motile cilia. There is a fundamental difference between the two. Primary cilia, non-motile. These are single, 1 per cell. They do not generate any movement. Their job is sensing, detecting physical and biochemical signals. They are mostly found on non-dividing epithelial cells. Gray's Anatomy describes them as the antenna of the cell, a very good analogy. Examples of primary cilia: in the collecting duct of the kidney, they sense urine flow. And, very importantly, the primary cilium is also the hub for the hedgehog signaling pathway. We will cover this in detail in a dedicated slide ahead. Motile cilia, these are present in large numbers on a single cell. They are found on the apical domain of epithelia. They beat in a wave-like motion. Their job is to generate fluid movement. Examples: they clear mucus in the upper respiratory tract. They move the egg in the oviducts, that is the fallopian tubes. They circulate cerebrospinal fluid in the brain ventricles. Now let us understand ciliogenesis, that is, how a cilium is actually built. It is a step-by-step process. Step 1: Basal body formation. The centriole-derived basal body migrates to the apical domain of the cell, meaning toward the top, where the cilium will form. Step 2: Transition zone. From the distal region of the basal body, that is, the upper part, axonal microtubule doublets emerge. This area is called the transition zone. It is the critical boundary between the basal body and the cilium shaft. Step 3: IFT complex is activated. Now the intraflagellar transport proteins are activated. These are bidirectional motor-driven proteins that import tubulin dimers into the cilium. Step 4: Elongation at the distal tip. Tubulin dimers are transported to the ciliary tip, and they are added there. This is how the cilium grows. Step 5: Steady-state length. Once the cilium reaches its final length, it stays constant. How? Through balance. A steady equilibrium is maintained between tubulin turnover and the addition of new dimers at the tip. One key interview point: the length of a cilium is not determined by a fixed gene, it is a dynamic balance. A very interesting concept. Now let us take a deep dive into IFT, because this is a concept where interview questions almost always come. The full name is intraflagellar transport. This is a bidirectional transport system, meaning it travels in both directions, from base to tip and from tip to base. There are two complexes, IFTA and IFTB. The IFTA complex works with the dynein 2 motor and performs retrograde transport, that is, from tip to base. The IFTB complex works with the kinesin 2 motor and performs anterograde transport, that is, from base to tip. An easy memory trick. A for away from tip. IFTA is retrograde. B for building the tip. IFTB is anterograde. Remember this. Now in the figure on the slide, you can see figure 1.26c and D. Part C shows axonal transport in a neuron. This is exactly like IFT, bidirectional. In anterograde, kinesin 2 carries vesicles, neurotransmitters, and tubulin toward the synapse. In retrograde, cytoplasmic dynein 2 brings recycled synaptic vesicles back to the soma. Part D shows the spermatids IMT, intramanchet transport. The manchet is a microtubule-affecting temporary structure that surrounds the elongating spermatid. IFT trains transport cargo along single microtubule tracks, kinesin motor in anterograde, dynein motor in retrograde. Now let us understand how cilia and flagella actually move. This mechanism is very elegant. Ciliary beating. The mechanism is this. Dynein arms have ADPase activity. They use ADP to slide the outer microtubule doublets over each other. This produces a large-scale bending motion. One critical point: the length of the microtubules does not change. Only their relative position to each other changes. Effective stroke. When the cilium pushes, it bends slightly at its base and the rest stays stiff. Just like an oar, the paddling motion of a boat. This pushes the fluid. Recovery stroke. Then to return to position, the bend travels from base to tip. This brings the cilium back to its starting position, ready for the next cycle. Pattern. The beating is planar, in one plane. But it is asymmetric between the effective and recovery strokes. Metachronal synchrony. This is a very important concept. A group of cilia beat in a coordinated way, one after another, creating long traveling waves. These waves travel across the tissue's surface in the same direction as the effective stroke. This is what makes mucus move smoothly along the airways. Flagellar movement. Flagella are slightly different. They move by rapid undulation, passing from the attached end to the free end. In human spermatozoa there is an additional helical component to this undulation. Flagella also have outer dense fibers and a fibrous sheath surrounding the axoneme. Now it is time for clinical application. This section is directly useful in both exams and interviews. Respiratory tract. Motile cilia sweep mucus and trapped particles out of the airways. This is called mucociliary clearance. If cilia fail, as in primary ciliary dyskinesia, It leads to chronic respiratory infections and bronchiectasis. Oviducts. Cilia move the egg from the ovary toward the uterus. A defect increases the risk of ectopic pregnancy. Brain ventricles, CSF, that is cerebrospinal fluid, is circulated by the cilia of ependymal cells. A defect can lead to hydrocephalus. Embryonic node. This is the most fascinating one. In the node of the developing embryo, cilia generate a unidirectional fluid flow, and this determines left-right visceral asymmetry. This is why our heart is on the left, and the liver is on the right. If this flow is disturbed, in Cardigan syndrome, situs inversus occurs. Organs are in a mirror image position. Retina and sensory cells. Modified cilia are present in photoreceptors, olfactory cells, and vestibular hair cells, for sensory function. Kidney collecting duct. Primary cilia sense urine flow and modulate duct morphogenesis. Polycystic kidney disease is linked to a ciliary defect. This slide is slightly advanced, but also very important. Gray's Anatomy directly cites 2 research papers on this topic, so we will cover those as well. Primary cilia are essential for the hedgehog signaling pathway. What does the hedgehog pathway do? It organizes the body plan, controls organogenesis, and if it gets dysregulated, it is involved in tumorigenesis. How does the pathway work, step by step? First, the hedgehog ligand, such as sonic hedgehog, binds to the receptor patched 1. Second, this releases the protein smoothened, also called SMO, from inhibition. Third, activated SMO migrates into the primary cilium. Fourth, GLI transcription factors are activated at the ciliary tip. Fifth, GLI enters the nucleus and activates target genes, which control the cell cycle, proliferation, and apoptosis. A mnemonic, patch SMO GLI. Patch releases SMO, SMO activates GLI. Simple. Now let us look at the research citations that Gray's Anatomy has mentioned. Briscoe J. and Theron P.P. 2013, Nature Reviews Molecular Cell Biology, Volume 14, pages 416 to 429. This paper was from the MRC National Institute for Medical Research, London. This is a landmark review that explains the full hedgehog signaling mechanism, patched, smoothened, GLI proteins, and the role of primary cilia in vertebrates. Wei G., Naslimova L. and Hancock J.D., 2018, Frontiers in Cell and Developmental Biology, Volume 6, Article 8. This paper was from the University of West of England, Bristol. This review describes the primary cilium as the cell's antenna, and covers all major signaling pathways that pass through cilia. Interview tip: if an examiner asks, "What is the connection between the hedgehog pathway and cilia?" you can confidently mention Briscoe and Theron 2013 by name. That is very impressive. Cilia are not just in the airways and oviducts, they are found in many different places in the body in modified forms. These modified cilia are very interesting. Olfactory cilia, found on the dendritic endings of olfactory receptor cells. These are chemosensory, specialized receptor proteins on the ciliary membrane detect smell. Essentially, when you are smelling something, these cilia are at work. Kinecilia, found in vestibular hair cells. These sense mechanical displacement for balance and spatial orientation. The function of the inner ear depends on these. Photoreceptor connecting cilium, found in the rods and cones of the retina. This is a highly modified form of primary cilium that connects the inner segment to the outer segment. Visual pigment proteins are transported through the cilium. A ciliary defect leads to retinal dystrophy and blindness, as seen in Bardet-Biedl syndrome. Nodal cilia, in the node of the developing embryo. These exist only very briefly but perform a critically important function. They generate a left-handed fluid flow that establishes left-right body asymmetry. These cilia rotate clockwise on the left side. This pushes fluid to the left, and that signal determines where each organ will be positioned. As we close, let us do a quick revision. These 8 questions can come directly in an exam or interview. Make sure you know all of them. Question 1. What is the arrangement of the axoneme? 9 plus 2. That is 9 outer microtubule doublets and 2 central microtubules. Question 2. Which motor drives anterograde IFT? Kinesin 2. Direction base to tip. Question 3. Which motor drives retrograde IFT? Cytoplasmic dynein 2. Direction tip to base. Question 4. How does a cilium bend? Dynein arms use ADPase activity to slide the outer doublets. The length of microtubules does not change. Question 5. What is the function of the primary cilium? Mechanosensory role plus it is the hub of the hedgehog signaling pathway. Question 6. What is metachronal synchrony? A coordinated wave of cilia that travels in the same direction as the effective stroke. Question 7. What is the role of cilia in the embryo? Determining left-right visceral asymmetry, cardiac situs, liver position. Question 8. What is the length of the spermatozoan flagellum? 70 micrometers. Movement, rapid undulation plus a helical component. So that was today's topic. If you found this video helpful, please like it, subscribe this channel so you never miss an important topic, and share it with your classmates.